Good morning, guys. It is Miss Manning, and today we're going to learn about DNA replication. We're going to learn how one DNA strand splits off and becomes two brand new ones. Seems intimidating. It's actually pretty simple. To start off, we'll review. Explain DNA's properties based on its structure. Double helix structure explains Chargaff's rule about base pairing. Nitrogenous bases are paired along the rungs of the twisted ladder. That is these bases here in the center. That's the base pairing. C and G always go together. A and T always go together. The two strands are anti-parallel. That means they point in opposite directions. This allows the nitrogenous bases to pair together like puzzle pieces. You can see in our picture here, the pentagons have their oxygen atom on the top, pointing upward. On the other side here, that same atom is pointing downward. Hydrogen bonds hold together the nitrogenous bases. Individually, these hydrogen bonds are weak and easily broken. But this property enables the molecule to unzip and be replicated easily. It's kind of like a strand of hair. One strand of hair is very easily broken, but a braid of hair altogether is very, very strong. Same idea is happening with our DNA strands. Here again is our picture of our DNA molecule. The pentagons on the left, that is the deoxyribose sugar, and that is pointing upwards here, downwards here. We have our phosphate groups over here on the ends as well, and they're upside down on the other side. In the center, we have the hydrogen bonds holding together all of our nitrogenous bases. Our bases are held together by these individually weak hydrogen bonds. Again, just a reminder that only certain bases will pair together, AT and GC. So let's get into DNA replication. DNA is replicated in the S phase of interphase, just before mitosis begins. We will study more about the cell cycle a little bit later this semester. Basically, what I want you to know out of this slide is that DNA is replicated just before mitosis begins. One strand of the helix can be used to create another new strand that is complementary. Each strand is a template for the new strands to build on. If you see here, my cat says hello. If you see here, these brand new strands are exact copies of each other. AT, 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 AT. The process of replication can begin at dozens or hundreds of locations in the eukaryotic chromosome. Eukaryotic, this part here, this is what we're talking about. It has a nucleus. The opposite of eukaryotic then is prokaryotic. Replication proceeds in both directions until each chromosome is completely copied. The process can begin at dozens or hundreds of locations. It will start similar to this. You see our first spot here. This is one of the first spots of our duplicating DNA strands, and it starts to get a little bit larger. But a second area here is also replicating. Both of those sections continue growing until eventually the entire strand, until these big bubbles have all joined in together. DNA helicase. This is an enzyme, which are proteins, and this enzyme unzips the double helix. exposing the bases by breaking apart their hydrogen bonds. Our helicase here, it is breaking apart all of our nitrogenous bases. This whole area here, where two strands come out of one strand. Our our original parent strand has split into two, and that split is happening at the replication fork. As I said earlier, each strand serves as a template for the attachment of complementary bases. These are the bases again. A will always match with T, and G always matches with C. With all of our exposed bases, we'll have two identical copies from our old strand. Helicase has split apart the base pairs here, and these two strands here at the bottom are identical copies. DNA polymerase is an enzyme that joins individual nucleotides together by connecting the sugar and phosphates with a covalent bond. DNA polymerase also proofreads the new DNA so that each molecule is a near-perfect copy. Now I say near-perfect because mistakes do happen, but polymerase usually proofreads and breaks apart bonds that shouldn't occur if, for example, A and C matched up. Polymerase would come along and make sure that doesn't actually happen. Make sure that those don't bond together. In class, we built the big DNA molecule. We knew we have the nitrogenous base here. The yellow piece was our sugar. The red piece here, this was the phosphate group. Now, here are little tiny green pieces. Those are the covalent bonds. And this will show you how and why, when we built the whole strand, these covalent bonds are connecting the phosphate group of one nucleotide connects to the next sugar of the next nucleotide. This is just a short segment of a DNA model that was created in class. And as you can see, we have all of our pieces here. We've got our hydrogen bonds holding together the two nitrogenous bases. The base is attached to a sugar and a phosphate group. And this whole piece right here is one nucleotide. This nucleotide here, this is just one piece, but if you look, we actually have another nucleotide right here and right here. And this includes all the nucleotides that are on the opposite side down here. They're just slightly more out of view. Now when we did our replication, we cut apart the DNA strands right here. So this was our splitting point. This is where helicase separated it. And this is where we were able to start bringing in exactly our next diagram. Again, that was the model that we were manipulating here in class. Mistakes in the DNA code can happen. Damaged DNA may be accidentally copied. This can result in alterations of certain genes. If you remember from the beginning of our unit, we talked about how UV rays can come in and disrupt all of our bases. The bases are really good at absorbing ultraviolet radiation from the sun. This is as simple as it gets for how a sunburn leads to skin cancers. What happens is that UV rays has come in and it will break apart these bonds, and maybe it would connect instead of A pairing with T. Maybe A does in fact pair with C, and then this accidentally gets copied in the next cell, and the next cell, and the next cell, and it never gets corrected. Well, if that's the case, and if this is happening within a gene sequence where the growth of the cell is being monitored, then your cells will grow incorrectly, and that's how a cancer would form. There's many types of cancers, of course, and this is just one of the methods that would cause it. Some mistakes can be kind of elaborate, some can be very simple, and some can be just no big deal at all, just completely unnoticeable. And sometimes it leads to X-Men and really cool mutations. That summarizes all of our DNA replications. I hope you guys have a great rest of the day. Come by if you have any questions. See you later.